Okay. Just making sure. Just give me 1 second. I'm just making sure it's getting recorded.
Yeah.
Okay. Yep. Okay. Uh, hello everyone. Welcome. Thank you so much for your time. And today our guest speaker for the webinar series is Dr. Lokesh Lingappa. Uh, Dr. Lingappa doesn't need any introduction. We all know all how involved he's in the CDKL5 community, all the great work he's been doing. He's one of the few doctors who saw a patient around 19 years ago. Uh, the child herself is 19 years ago. So with all his experience, we are great to have him on board. And he recently attended the CDKL5 conference in Boston, and he's going to share all the updates and all the updates from there, as well as talk about clinical and research.
Yeah. Thank you, Jainu, for that kind introduction. Yeah.
Yes.
So we can go to the next slide. Yeah. We can move to the next slide.
Yeah, yeah. I think there might be a little delay, but do you see the John Howard slide on your?
So this is held in a city where Harvard University is there. So it was a pride moment for me to represent the Southeast Asia CDKL5 community. And next slide, please. Yeah. So just to see what is the disease expectation of parents, what is the scientific progress, where do we stand today, and what is the future ahead? We'll just to cover few of these points as we go along. I'll probably mute this just a second. Yeah. So this is one of the poster we presented, and among the 20 posters which were presented in the meeting, only 2 were the clinical papers, this paper along with the paper from Turkey. Rest of all were actually based on the basic science work which were being going on, and some of the drug trials and the natural history studies. So we had around 18 children in our follow-up who had been genetically proven this one with the average age of around 47 months, starting from 3 months of age to 171 months. So we had a good follow-up. Only one of the children had died due to pneumonia at 3 years of age. As you can see, most of them were significantly disabled, and 4 of them were able to walk actually, 2 without assistance and 2 with some assistance, minimal assistance. So of the 18, the 17 who were alive, around 4 were ambulant. So there is these things, and on the communication side, word output was very limited, and only one child had around 2 to 6 words, and other child had just a little bit of pointing, and almost 16 of them didn't have any vocabulary or significant understanding. So that is what our experience of these 18 children were. And the seizures were almost every one of them had seizures, and multiple types of seizures were there, and multiple medications were being used, including the cannabis oil and also the ketogenic diet in some of them. So it is quite challenging disorder, and we are increasing our number of enrollment, which is the way to go to do any further studies. Next one, please. So how does CDKL5 cause the epileptic encephalopathy and all this developmental this one? So there are multiple mechanisms. Not everything is understood. I don't think so. We need to go into detail here. So one common thing is it has presence in the cytoplasm and presence in the nucleus as well. So it has multiple actions. It also interface with the Rett syndrome gene as well. So MECP2 is the Rett syndrome gene. So that's why there are a lot of similarities and differences between the two disease mechanisms. Next one. Next slide, please. Yeah. So this is just one of the old study which says that depending on the genetic mutation, your chance of walking increases significantly. If you have mutations on exon 12 to 21, your chance of walking is much better. So this is one of the study, although it is an old study, but I think this is one of the landmark studies which increased the hope of the CDKL5 community. Basically, what they found is if you activate you can turn it off or turn off the gene in the mouse, and they could see you can actually restore all the disease symptoms even if you restore the gene function in the adulthood. Next one, please. So which is very, very difficult in most of the other developmental disorder. That's why CDKL5 is slightly unusual, that you can actually reverse most of the symptoms. Next one. I think that gives a significant hope. So this is one of the key point where CDKL deficiency looks like a neuronal maintenance of the ongoing function of the brain as compared to neuronal development. So what the difference would be is, if something is necessary for neuronal development, after 3 years of age, you will not be able to make much of difference, because the most of the development is in first 36 months of life, and later on very slow development in next 8 to 10 years. But if it is a maintenance gene, you can alter it and it can reverse a lot of symptoms, and that is what very hopeful for the CDD, what it is at this point in time. So there are a lot of questions still, but I think this is one of the landmark study which gave us a lot of understanding and input. Next one. Yeah. So what they, yeah, the next slide. So they, what they did was, they, even at the later age, when they restored the CDKL5 expression, it rescued most of the CDD deficits in the mice model. So which is exactly, which is one of the very important thing. And also they stopped the seizures as well. So they measured in multiple different ways and made sure they are measuring the seizures, because it's not easy to measure a seizure in the mice model. Next one. So I think at the biological level, in this how it functions more like a channel abnormality rather than a developmental gene or a structural gene abnormality. That's why we are able to find the degree of reversibility is what was the outcome of this study. Next one. So the opening talk in the meeting was a very impressive talk by a sibling, and what she shared was how it impacts other kids at home. There is changing quality time of to the normal sibling. That's one of the important thing. Isolation from the community, because parents are traveling to hospital, either admission or the consultations, taking care of the child who requires 24/7 care. So that causes significant isolation from community. But at the same time, you develop more empathy, you are more compassionate to your peer group, because you are going through so much at home. So it makes you a different individual. That what shared. I think that we have seen in many of these families. And one of the important thing they would appreciate is open communication about the time and care needed for these children with any kind of developmental disabilities. So this is what we can do, and this is what we are unable to do to you. So that open communication probably makes a huge difference. And a family staying together is one of the single most important thing they appreciate. In spite of all these difficulties, I think that is one thing which will hold them together, and then have a sense of belonging, and they help in the care, supportive care. The next one, please. Yeah, I think this is a 2015 slide, which where it all started, the CDKL5 therapies. All the different angles were looked into and earmarked as to what all areas we need to work on. And there are a lot of progress which has happened in the last 10 years. Next one. So there is a saying that if to get anything new out of a research, you need to have a technique to research it. First and foremost, you need to be passionate about what you are doing, and you need to have new ideas in that order to succeed in any research. So that is very, so if you don't have a technique, even if you are passionate, you have new ideas, you cannot test your hypothesis. So that is why all these things are evolving so rapidly. So we are hopefully closer to what we want to do. Next one. So I think there are certain things which are not probably required as a parent. I'll just go through what are is involved to understand this. So if I am putting in CDKL5 into a mice model or into a organoid, I want to study that how much of CDKL5 I'm able to generate. So you need to have what is called as antibodies, a tagged antibodies. They will have a fluorescent thing which will light it up. So if there is antibody which is developing, or if there is a CDKL5 protein which is being produced, and you want to test in the mice in vitro, in the brain of that mouse or on the organoid. So if you put in a fluorescent antibody, you will be immediately able to check on your scan saying that, okay, these are all the areas where the CDKL5 has come up. So that is why people are trying to develop the antibodies so that you can easily measure the effect of the therapy what you are giving. Next slide, please. So we'll just quickly go through molecular biology. So that is not CDKL5 is not the only one what is called as isoform. So there are total 1 to 5 isoforms. CDKL2 is another variant. Rarely it causes growth failure, some intellectual delay or seizures. CDKL1 rarely can cause global developmental delay. But generally, they are very tolerant to any gene variation, CDKL1 to 4. But CDKL5 is very intolerant to variation of the genetic content. So that is why it causes the disease and significant long-term impact. Next slide, please. So I think we'll skip all this. So it is just to show you can rescue the CDKL5 by increasing the expression of CDKL2. So it causes reduced seizures and also improved mobility. So what they have found is if you increase the ability to express the CDKL2, it can rescue some of the deficiency of the CDKL5. So this is one of the mechanisms which people are trying to work on to try and improve the functional at CDD. Next one. Yeah, next one, please. So I think I'll skip this. Yeah, in the interest. So another thing what we will have to also know is when you are trying to express this CDKL5 or improve the by a genetic thing, you are trying to increase the expression of CDKL5. You also have to need to know what is the dosage that is just enough, what is the dose which can cause more toxicity. So that is one of the important thing you need to be aware of, because what happens is if it is too much of a dosage, whether it can cause more some other disease, because there are conditions where excessive expression also causes disease, reduction in the CDD is causing a disease. So that is we need to understand, and generally what I so far is understood is CDKL5 excessive expression is not so toxic. Next one, please. So I think there is a video. Yeah. So these are the videos of flies which shows if the CDKL5, this one is there, their ability to, they're all having some seizures, and some of them, the ability to climb up on the wall were significantly reduced when they had a CDD. So you can see they had climbed up quite well in the normal ones, wild variants, whereas the diseased ones had a problem. Yeah, the next one. Yeah, next slide, please. Yeah. So whenever you rescue the CDKL5 phenotype by gene therapy or anything, there is a significant improvement in all these phenotypes. Yeah, next slide, please. Yeah. So CDKL5, yeah, we can keep be here. So the next thing what one of the study from Cambridge was cortical neuronal quantification for design of CDD gene replacement. So where all you need to look out for this CDKL5. So you need to look out for in the nucleus, you need to look out for the cytoplasm and the whole cell as well. You need to count in different areas, like you have to count in either the thalamus or the cortical neurons in the occipital cortex, all the different areas you need to look at to quantify these neurons. Next slide. I think we will skip this, it will be. So, as we all know, the incidence is around 140 to 60,000, and in UK they detect around 14 EDD in a year, and they are trying to do the precise CDKL5 expression analysis to match the CD gene therapy to the baseline levels in the normal children. So they are trying to see what will be the amount of gene which actually has to be expressed to make sure you are not causing any more problems. Next slide. Yeah, next slide, please. It's all just, these are all studies. I'm not trying to say we need to know all of these things. How much of research actually is going on to
Uh, I think Dr. Lokesh got disconnected.
yeah, got disconnected. I don't know.
Okay, you back? Yes.
Yeah. So I'll just try to why we are trying to go through all this. These are all the things which are actually present at meeting. The need for these kind of basic science work is to get a three-dimensional view of where is the defect, what is the quantification of it. When I put in some therapy back in, how am
Uh, Sir, we cannot hear you. Lokesh, sir. So basically, what Dr. Lokesh sir was saying is that this is a therapeutic tool that has been developed in Italy. And what this does is it talks about these different disorders and it shows that for these different rare disorders on the left hand side, at what stages we are, like Boston Children's Hospital, do we have iPSC assays, do we have in vivo assays for that, and do we have therapeutic studies for that? And as you can see, like for CDKL5 specifically, most of these are already been present. Yes, sir. Welcome back.
yeah, yeah.
Okay.
Yeah. Sorry, there is some glitch. Yeah.
Definitely.
So I think that slide has been already explained by Jainu. That is what we are looking at. So there are multiple disorders like this. People are working together on multiple disorders. And one of the concept, yeah, you can go to the next slide. So one of the yeah, next slide, please. So one of the concept you need to understand today to understand the disease is what is called as induced pluripotential stem cells. So you are able to have some enzymes and the gene directions. For you get a cell and then which is a stem cell, and try and induce them in a different direction to create a cell of a neuron of a particular area, and later on you can form an organ like, there is a basal ganglia, there is a thalamus or a hypothalamus. So the scientists are able to create a organoid, a structurally connected brain part in a petri dish. The beauty of it is, so you have all the chemicals which are forming in the brain in a petri dish, and it is functional. So then into that organoid you can actually introduce, you have one normal organoid wild, you can take a gene from the deceased adult or child and then put it into the organoid. So it will develop the disease manifestation, which can be studied with the EEG, which electrical this one, and how the motility movements of the cells, all those things can be studied in a petri dish, which has become one of the gold standard along with the mouse models or other things which are zebrafish models, all those things which are being used. This has become one of the gold standard in last 4 to 5 years. Next slide, please. Yeah, I think we'll skip this. Yeah. And one of the paper which was presented is whether there is a autophagy, that is increased self-destruction and neuroinflammation to battle against the virus. That is one of the paper which was being presented. Next slide, please. So I think I'll not go into too much of detail. There has been found that there is a slightly increased incidence of viral infections, especially something called Sinbid virus, which is usually Sindbis virus, which is usually found in Africa. So that is a good model to say how this children with the CDD versus without CDD, how they get affected. And there is some role in the immunity as well, which has been shown with this Sindbis viral model. Next one, please. Yeah, I think we'll skip this. Okay. So one of the sessions from King's College London was increasing kinase activity in CDKL5. Can it reduce the effect of CDD? That is, in absence of CDKL5, there is a phosphorylation is a process which happens in the cell. It is around 15 to 20% which is seen. Whereas in a normal this one, it is around 90 to 100% phosphorylation. Next slide. So they have looked at various forms to see how the phosphorylation happens in different parts of the brain using the mouse model wild type and CDKL knockout mice and CDKL5 and CDKL2 knockout mouse, and they have found various information on to see that the phosphorylation is significantly reduced if you have CDKL5 deficiency. Yeah, next slide, please. Yeah, no, you can skip this. Yeah, thank you. Yeah, same thing. We can skip. Yeah, this one as well. So there was one study from Toronto wherein they taken only children with exon 19 mutation, 11 children globally, and this was seen in around 12 to 23% of the data from the Boston, sorry, not DCH, it's a BCH at Boston Children's Hospital. On of the 300 children, it constituted around 12 to 23% of this gene, and they studied specifically this gene mutation. Next one. Next one, please. Yeah. Yeah. So what they looked at is there is a CDKL5 serine threonine kinase target, which actually has two different proteins, and you can actually change the phosphorylation by using a injection. Next one. This is the injection ixozimib, which actually is used in cancer chemotherapy and other conditions, and it is called what is called as proteasome inhibitor, and is clinically approved for other indications including malignancy treatment. Next one, please. So they have given treatment to hemizygously deleted control this one mice models, and what they have looked at is whether it improves the behavioral phenotypes and does it improve the biomarkers. Next one, please. Yeah, next one. So what they have clearly seen is when there are significantly increased electrographic seizures, generalized seizures, and status epilepticus in the heterozygous model. Next one. So when you had given the drug, there is a significant decrease in the electrographic seizure, which was noticed almost 50% decrease in the electrographic seizures, which was noted when this drug ixazomib was given. So this is all experimental models to see whether immunomodulation and these kind of things to improve the phosphorylation will actually change the clinical phenotype. Next one, please. Yeah, next one. So it is showing the same information. I think this one I'll skip. Yeah, this one as well. It says about the exosome. Yeah. So induced pluripotential stem cells and collaboration. This is one of the important thing. People are trying to collaborate and have a standardized form of induced pluripotential stem cells, which can be used across the research. So you have one laboratory which has standardized how to produce a CDD stem cell line, and then that can be used for a one research across different centers so that you have a reproducible model. So that is why they are trying to standardize this, and so that all the CRISPR technology or Cas9 patient cell lines can be standardized so that your research outcomes are similar. Next one. Yeah. So this is another study to look at where is the problem when the vision is impaired. So what they found is it is not in the thalamocortical circuit. It is more likely to be in the circuit which is before the thalamocortical. That is when the impulse is reaching the eye to the nerve, then nerve to the lateral geniculate body. So the problem is at that level, whereas the thalamus to cortical circuit is preserved. So that is what the one of the study looking at the why the visual impairment is there in these children. Yeah. Next one. Yeah, I think this is the same information. Thank you. They also have increased tactile sensitivity and tactile reactivity as well, because these somatosensory neurons also contain the CDKL5 gene. Next one, please. The same thing, dorsal root ganglia are affected, which is on the spinal cord, and they are highly expressed in the sensory ganglia. That's why you can have even the colon might not be functioning well, and they can have altered pain response to the any like abdominal distension or the colonic distension. They can have a pain response which is altered, and unusual irritability can be there. Next one. Yeah, I think we'll skip. Yeah. So another studies from the Jackson lab is one which produces a lot of mice models to mice and rat models. They are also trying to standardize so that it can be used for different studies, including the CDKL5. So they have a standardized wild type and also the disease models, so that you can do your experiments in their lab. Was that what they were proposing? Next one. Yeah, I think we'll skip this. Yeah. So second day was more about what are the different options are there, therapy options, which probably will interest you more. Yeah, then you can go to this first. Sir Colin Blackmore was the person who was the scientific advisory person in the initial period, and who unfortunately died of, I think, the motor neuron disease, and he had done significant work to improve the scientific community getting together for this condition. So what are the therapies available already in different models? So intrathecal enzyme replacement therapy, that is giving into the brain, has been seen in multiple conditions as of now. So this is for MPS type 1, that is mucopolysaccharidosis type 1 canine model. After intrathecal enzyme replacement therapy, they had found there is a significant efficacy improvement by more than 1,000 times as compared to giving something intravenously. So that giving IV versus giving directly into the brain, there is almost 1,000 times more improvement, because you are crossing the blood-brain barrier directly. Next one. So that is one thing to say, delivery of the drug, how to deliver the drug. Okay. So this is one condition called late infantile Batten disease, or CLN2 gene defect, which causes significant defect, rapid iteration and death. So as you can see, the control without intervention, they die quite rapidly within 10 to this is in the weeks in the mouse model, which rapidly decline. Whereas once you treat, as you can see, they are very well stabilized for long period. So that is a very good evidence, and it is approved, FDA approved, and it is available as a treatment option in European community and also in the US. But it has to be given to the brain directly, intracerebroventricularly. Next one, please. Yeah. So this is for the Angelman. There is a lot of trials which is going on. GTX-102 is one of the thing which is used for Angelman disease. Different follow-ups have been looked into cognition, receptive communication. Next one. Yeah. So they have seen the treated patients clinically showed significant improvement in all the aspects of response up till almost 2 years. And as compared to natural history, there was a significant and continued improvement beyond 1 to 2 years once these children with Angelman were treated with this drug called GTX-102, which is still in the enrolling the phase 3 trial as of now. Next one. Yeah, I think showing the similar kind of thing, we can skip this. So Bailey score and all the other ones. So a lot of different information have been looked into, and it all showed that there is a improvement in different aspects. Irritability, behavior, cognition, everything was better for the Angelman, this one. As compared to what they have found is Angelman, you cannot reverse most of this one. Only part of the problem can be reversed at this point in time. So that is what happens. Whereas in CDD, as of now, with the animal model, what we have, we are hoping that we can reverse it completely. So that's the difference between the Angelman versus the CDD. Next one, please. Yeah, I think we'll skip this. This is all about the Angelman. Yeah. So UX055 for CDKL5 deficiency disorder. So this is the efficacy of the genetic treatment, what they are trying to do. And they're looking at the expression and efficacy in CDD patients
And this
using the
one is by Ultragenyx,
iPSC-derived
right?
neurons.
This one is by Ultragenyx, right, sir?
Ultragenyx, yeah, Ultragenyx, yeah. That's Ultragenyx study, yeah. I think I have a little bit better slide there later on. We'll go through that. Yeah. So one of the important thing is how do you, yeah, we can go to the previous slide. How do you make sure that it is distributed in the brain and it goes into the neuronal transduction and it expresses the gene across a large brain area. Normally we are studying in the mouse model or mice model, which is very small surface area. Whereas when it has to be transferred into a human brain, you need to have big brain models which shows, okay, if you deliver a drug in a particular manner, it's going to reach to the different parts. That is one of the challenges we need to, well, are extrapolating the problem from the results from the mice model to the human model. So that is one of the calculations you need to do, and maybe you need to do it in a pig or something else, then okay, larger side brain, then do in the human models, so that you are ensuring that the dose and the distribution and the transgene expression, everything happens in an appropriate manner. Next one, please. Yeah, so this is just a how it is being done. So intracerebroventricular injection. So it is into the ventricles in the brain. They are given at 3 to 5 weeks of age, and so what they have seen is high levels of freezing, which is associated with the successful learning. So if you make them sit at one place, because they are generally very hyperactive running around. So if the gene is effective, gene therapy is effective, they are calmer, they sit at a place, they will have to have better hind limb clasping, all those things can be seen, and they reverse lot of symptoms. Next one, please. Yeah. So, and then the second target, the issues what they are looking at is what is the optimal dosing and use of neuron specific promoters, because you can just adeno associated virus, I think most of you are aware, is the usually vehicle in which the gene is kept in, but along with that you have to have a promoter which can help you to attach to the particular area and make it to translate there. So what they have found is, if you are doing a lumbar puncture, Trendelenburg position is helpful. Low volume flush is required. And yeah. Yeah. So intracerebroventricular. Yeah. So the intracerebroventricular, this just previous slide, if you can go, sorry. Yeah. So as compared to lumbar puncture, what they have found is in the cervicomedullary junction. So it is the neck and the spine junction. So if you give the injection there, the delivery is much better, is what they have seen. So that is another improvement in the technique of delivery of a gene. So you are not running from just one, you are running different experience of different genes which are already in multiple phases, advanced phases. So now technique wise, either you give intracerebral ventricularly, or which is more difficult in some children. So otherwise you can give to the cervicomedullary junction. It has to be given generally by a neurosurgeon. That has a better delivery of the drug. Next one, please.
I have a quick question here. So in the mice model, they did ICV, but then
Yeah.
they're talking about ICM and lumbar. Uh, so what is the one?
Yeah, no, this is actually the human models,
Uh-huh.
this one where they are actually human and the monkey models. What they are trying to say is the intra isolary as compared to the lumbar puncture. But that ultragenics, this one they have not tried in this thing.
Okay, so this is just this is better occurred in humans or monkey
Yeah,
models
it
that we've
just a better
seen, but
delivery. Yeah, better
yeah,
delivery in
but
the
we've not
humans.
tested
Yeah, yeah.
the specific the ultragenics one. Okay, thanks. Yeah.
First next this one. Yes, exactly. Yeah. Next one, please. Yeah. So after the procedure, keeping in the Trendelenburg position is shown to improve the delivery of the drug, that is lying down, down this one. Next one. Yeah. So I think just put in the, yeah, next slide, please. In different areas, they have shown the same thing. I think we've gone through this already. So I think this is once again the adeno AAV virus for CDD, which I think this slide has come in between here. So this Angelman, this one, they are also using anti-ASO, antisense oligonucleotides to alter the gene expression. So this one, which is in phase where they are almost trying the phase 3 trials. Next. Yeah. So I think a lot of you are aware of the Candid study, which started a few years back. So basically, what they are trying to do is, so if you have to be ready for a trial of a drug. So for that, you need to have, one is natural history across different ages, how it progresses or whether it remains steady. So that's why they taken all ages, split it to 0 to 2 to 5 and 6 to 12 and 12 and above, like that different age groups, they are collecting different data. And this is one of the study where all the sort of companies are also, 7 companies are also part of this study to get that natural history standardized, and they are looking at all these scales in a standardized manner. Next one, please. Yeah. So they're trying to look at what is the change in test results over time in 6 months and other say 36 months, and correlation between the age and the test results, and between the gender and test results, is there a difference? Test result and seizure severity, is there a difference? So all this we measured, this is being ongoingly measured so that we have a baseline data. So if we can try, and as a parent community, we can do it wherever we are. We can try and collect this data on a prospective basis. We don't have to be necessarily part of the study, but at least if we collect this information, it will be greatly helpful when the study is there to come. We have the baseline data to show them. Next one. I think I'll skip this. It is about, once again, outcome measures. So yeah, so this is fine. So there are drugs which are being used to treat the seizures. One is ganaxolone, as you are all aware. This is a first approved drug for treating the seizures in the CDD. All the other drugs which are on the different phase.
I think we lost Dr. Lokesh again. So as Dr. Lokesh was saying, ganaxolone is already US FDA approved for CDD. Altalurin is specific to missense mutation, so it's not used for all the children. Takeda, this is a Takeda drug, Takeda 395, soticlestat. This is still in trials, and fenfluramine is a repurposed drug from
Is there a problem
obesity.
on my end or
Yes, sir. I thought like we couldn't
hello?
hear you, so if you're back, then
Hello?
can you hear us? Can you hear me?
Yeah, no, yeah. Actually, I don't know why it is getting cut. Yeah,
No problem,
yeah,
sir. So, yeah, so you can continue here.
yeah, yeah. Sure. So, while the slides are being loaded, uh, uh, the uh, uh, the next the adeno-associated virus ultragenics model, they are trying it on the organoid. So as I already explained about the organoid, so they are trying to do predominantly the organoid models, and also they are extrapolating that later on into the mice model. So both of them are going simultaneously. I'm still not able to see the screen.
So we said we are on the the different options available today, where you had ganaxolone, Altalurin, Ovid, Takeda 395, that is soticlestat, and fenfluramine. That's the slide we are still on. I'm not sure why it's not loading on your end.
Yeah. Yeah, I don't know. It's not loading on my end. The connection is okay. I'll
Mhm.
stop my video. Maybe that might help.
Okay.
Yeah, you can move the slide and you can tell me. I can just
Yeah. Mhm.
keep talking.
Sure. So the next slide we have is
Yeah.
gene replacement and X reactivation.
Yeah, X reactivation. Yeah. So since it's a predominantly X-linked disorder, so one of the thing, this one is the where there is a uh, you can actually do a epigenetic editing and then increase the expression of the silent X which is there. So that is what they have tried. So this is you can go to the next slide. Uh, I'll just see whether it is seen on this one for me. Yeah, the next one. So what they have looked at is from UCSF, University of California, optimized and validated dual adenovirus approach to deliver novel epigenetic editors. So one is you just put in the gene and send it. Here they are putting in the epigenetic editors to reactivate the healthy and the silent copy of CDKL5 to treat girls with CDKL5 deficiency. There are a lot of silent copies on the X gene. So they are trying to reactivate those healthy and silent copies so that it can come to rescue of this one. So what they have seen in brain organoid model is if you rescue by 70% of that activity, you can actually have a normal levels of functioning in the neurons. So you don't have to have 100% rescue. Even if you rescue 70%, you can actually have a good rescue and good activities that they have checked with the neuronal function as measured by the microelectrode arrays. Next one, please. So one is delivering the gene and the other one is this way. So they are also validated from the organoid into a mouse therapeutic model in a transgenic mouse model. Following AAV9 delivery into the CSF space, they were able to see near complete rescue and cognitive performance. So this data, they are trying to use it for fund application for human studies. Next one, please. So ultragenics, I think this is what I was saying earlier. This is adeno-associated virus vector and which contains a neuron-specific promoter intended to drive expression of functional CDKL5 specifically into the neurons. And it is delivered as a CSF through the CSF as we saw earlier into the cerebrovascular regions. And what they have found is it showed good improvement in motor and cognitive behavior empowerment in the mouse model and also led to correction of neuronal hyperexcitability in the human organoid model. So everything now they are doing parallelly. You do it in a mouse model and also you do it in a human brain organoid model. Next one, please. Yeah, I think things which we have discussed earlier. Yeah, next one. So, and their next step they are looking at is what is the minimally effective dosage. So you can give multiple dosages and study what gives the optimal effect, what gives any side effect. So there are what is called as dose finding study. In other apart from this one, even in the uh, regular drugs like anti-seizure medications or some other medication, we need to find what is the optimal dosage. So they try multiple dosages. So similarly, the dosage of the gene also, they will see multiple variables and see, okay, this dosage is perfect enough for us to
Uh, Lokesh, sir, we cannot hear you again.
yeah, I know it's just some reason this has happened. Uh, okay, I think we are almost at the last slide. Uh, yeah, yeah, dose finding study, and then after that we have, I think, only at least we can come to the summary slide so that we can have discussion. So before I wind up that, on our end, we have the study approved for the usage repurposing of allopurinol, and so that study as we're going to start enrolling for that study, and we'll shortly publish send this one details on the enrollment process and this one on the CDKL South Asia website. Jaino, you want to add anything on that?
Yeah, so this is the CDKL2 medicine that Dr. Lokesh is mentioning. And as you see that CDKL2, as Dr. Lokesh explained, CDKL2 has overlap in the phosphorylation targets as CDKL5, as we were talking earlier about that. And we got the CDSCO, which is the Indian equivalent of the FDA. We got their approval earlier last month. So we have the protocol, we have the IRB approval now. So all those steps are in place. And as Dr. Lokesh mentioned, the next steps from here would be to inform the community how to enroll the children and what would be the steps, what would be the testing that is needed before and during this clinical trial. Most of this will be
Yeah,
done at Rainbow
I,
Children's
I think
Hospital.
that. Yeah.
Mhm. Most of this will be done at Rainbow
Yeah, I
Children's
think what
Hospital.
we have is we also have network of children's hospitals. So if there are people which is closer by, we'll get approval of the study from once this is a primary approval site, we can make it multi-centric.
Yeah.
And maybe others people are also having larger, will try and collect from the clinicians. If there are sizable numbers more than 5 at a given center, we can ask them to get a local ethics committee approval, and you can have a multi-centric study simultaneously. That's what I'm planning to do.
Yes, thank you, sir. So basically, the primary site, as Dr. Lokesh mentioned, is Hyderabad. But as he's saying that you don't have to travel to
Yeah.
Hyderabad because it will be done at multiple children's hospitals. So if you can, like do it locally, that it will be ideal for the family, and that will be
Yeah.
we'll be
So
very
yeah,
grateful
that
for the
rolling out
patients.
might take a little bit time.
Okay, yes. So yeah,
Yeah,
we
the
can take
rolling
this
out
up.
might take a time because each individuals. Yeah.
Yes.
Yeah.
So
Any questions? Any this one?
let's see. You can put the questions in the chat window, or you
Sorry.
can, yeah. Uh, or
Yeah.
you can, raise your hand also, or you can unmute and ask the question. We're towards the end of the conference or the webinar. So, this is a good point for question and answers. Also, we got the ODD, Orphan Drug Designation approval in the US FDA for the same small
Okay.
molecule. Yeah.
Okay, that's good. Okay, I think we can use that information actually.
Yes.
Yeah.
So, yeah, I see. Yeah, I'll read the question to you, sir, and then
Yeah.
okay, so Vineil has a question, which is, is gene therapy possible in India once it gets approved in the US? So how would that work?
Absolutely. Yeah, absolutely possible because we are doing gene therapy in our center for spinal muscular atrophy. So which is one of the most popularly used gene therapy around the world. And So it is essentially we have everything to do it at India. So once it is approved and it is available to be shipped and all the clearances are there, so in place we can do it.
Mhm.
The
And
only thing
will
is
it be
most of these things are cost prohibitive. Yeah,
will
sorry.
that be will that be simultaneous or will that be
No, no, it will never be simultaneous. At least it will take at least 1 to 2 years from the time, unless we become the study centers for these diseases, for these trials, then we can. So I think that's why we collecting the data as what the candidate study is doing on all our patients, having those things ready would make a good point to these trial centers to say, yes, we have this information as you have in other centers, we can be centers for the trial.
Mhm. Thank you, sir. So the next question is, when will these trials be available, and will these trials be open to boys as well? So I'm thinking he's specifically asking about the gene therapy,
Yeah.
and yeah. So will it be available to the boys as well, and when will they be available
Yeah,
for
I think
human
it
trial?
is. Yeah. If it's too early to say when it will be available, because we are in the mice model and the organoid model level. So next they'll have to go to a bigger animal level and then see how the distribution works, what dosages, and once that is done, then we can actually look at rolling out into the human trials. And yes, it will be open to the boys because it's a gene therapy. There is no differentiation. It will be applicable to the boys as well.
Mhm. So there are other treatment options also that I wanted to add, and like protein replacement therapy or the CDKL2 target, those
Yeah,
definitely are available for boys as well as girls. They're not
yeah,
specific to maybe perhaps X reactivation. That one
yeah.
may
That
be
will not be
specific.
available.
Yeah, that is
Yeah,
specific to girls only. So
yeah.
there are, depending on what the modality of treatment is, they will be available depending on that. Yeah.
Yeah, that's the only thing which will not be available for boys. That's a X-ray activation, epigenetic modification. That's the only thing. Rest of the things should be common for both boys and girls. That's a
Yeah.
good point.
So, are there any other questions? You guys have Dr. Lokesh here, like please make use of this time.
Yeah.
He's very difficult to get hold of otherwise. Yeah.
So I just pasted one question.
Yeah.
Uh, Sorry, did I miss it? Okay, let me check.
What would be the estimated cost of gene therapy?
Okay, the estimated cost of gene therapy. So
Yeah.
basically in the US, it is approximately $1.2 million per person. That is the biggest challenge to get anything to India. But as and when we have more patients and more information and more data and the technology gets cheaper, so I think the SMA, sir, correct me if I'm wrong, it was around
Yeah.
16
Yeah.
crores,
16 crores. Yeah. 16
16
crores.
crores. Yeah.
Yeah.
So uh, this gene therapy also is along the similar ballpark numbers. Uh,
Yeah.
it is
It
going to
will be
be,
similar.
yep, it's a very uphill battle, but definitely this is being communicated to our industry partners, and we are working on getting the cost reduced.
Yeah. Because for Duchenne's muscular dystrophy, it is 24 crores.
Wow. Okay.
Yeah.
So, okay, the next question is, uh, can we talk a little bit more about the CDKL2 medicine and the dosage? So that is part of the protocol itself, and
Trial.
Dr.
Yeah.
Lokesh will explain that in more detail. Please go ahead,
Yeah.
sir.
Yeah. So the medication is sort of allopurinol, which is used to reduce the uric acid, and it is also known to improve the phosphorylation of the CDKL2. As we discussed earlier, CDKL2 increased expression or increased phosphorylation is known to partly rescue the phenotype of the CDKL5 CDD. So
And it's
that is what
around
we are trying.
33%. Like what we know from theoretical research is around it will
Theoretical.
rescue
Yeah.
33%
Yeah.
overlap. So it will rescue like 33% problem solving. It's not
Yeah.
100% problem solving, unlike gene
Yeah.
therapy or some other treatment options.
Yeah.
So the next question is, I have been trying to make an appointment with you, Dr. Lokesh,
I think
but
that you can mail me or message me. We can discuss later. Yeah.
yes, so I've heard that from many people. Like there is some issues in, I think, in the Rainbow
I think
Children
what you can do is
Hospital.
you can share my number. They can just message their name and number. We'll try to
Okay.
facilitate. Yeah.
Okay, I'll put it in the WhatsApp group one more time. Thank
Group
you
so
so
that they
much.
can message me. Yeah. With their
Yes.
number so that I'll facilitate. Yeah.
Yes. Yeah. Okay. Let's see. Any other questions?
Hi, January Suhasini here. Sorry to
Hi,
interrupt
Samson.
in between.
No problem.
We
Please
No
have
go
problem.
also
ahead with the
tried to
question.
that number what you have put in the group. We have no response actually, Lokesh sir. So we have
Which
taken
one?
an
No, no, I
appointment.
am asking to put my personal number.
Okay.
And then you just message me. I will facilitate. Okay.
Mhm.
For CDKL2, we have taken already appointment 2 months back for CDKL2 medicine. So
Okay. Sure.
me
No,
and
no,
one of the other
it's
friend.
a video consultation, is it? Yeah.
Yes, sir.
Yeah. Sure. Just message me again. I'll from tomorrow we'll try to sort it out. Yeah.
Sure.
Thank you, sir. Thank you so much for
Thank you.
that. So the next question is, does the climate impact CDD seizure pattern, especially during the uh, cold
Yeah.
weather? Yeah.
Yeah. So any of these stressors like a poor sleep because of cold, any increased body temperatures, and some of the medications like antihistamines at a higher dosages can all increase the seizures, not necessarily in CDD, but in general. So those factors might increase the seizures even in CDD.
Mhm. Yeah. So basically, like CDKL5 is also known as the Goldilocks of disorders, meaning that you need
Yeah,
to have the perfect balance for everything. So if
yeah,
it becomes
absolutely.
little hotter or little colder, then the seizures have been like there has been patients who have reported that the seizures had increased. Yeah,
Yeah.
that is. Any other questions?
So is that CDKL2 is ready to use for the
Yeah.
babies right now? So do we consult Prashanti ma'am as well for the medicine?
Yeah, in a sense, trial as a part of a trial, we'll start it because we get gather the data. So we can, the drug is a very simple drug, very cheap, and this one. But because while we are doing this, it is better to have a data from our own community, because that will be they what we cannot use it to repurpose the drug, because what they said is you can use it to prescribe as a investigator initiated study of the CDS score what it has told. But you will be able to easily prescribe this, and it is very doesn't have much of side effects. And blood test, yes, we have a list of blood tests. Jainam can explain in detail what has been suggested.
Mhm. Yeah. So, thank you, sir. So, as Dr. Lokesh mentioned, this is a repurposed drug, which means that it is not yet approved for CDKL5. However, since it's already FDA approved and this medicine has been known for its safety profile, as Dr. Lokesh said, it is a very simple medicine. So, it's known for its safety profile since the 1970s. And repurposing meaning that we are trying to use it for a new drug. So it's been approved for the clinical trial that you can participate in the clinical trial and give it to the children, but it's not approved for prescription yet for CDKL5. And there are blood work that needs to be done, especially with respect to measuring the uric acid levels, because this is a gout medicine traditionally, and it impacts the gout, the uric level. So as a side effect, we don't want certain levels to reduce further down. So that's why there will be blood panel, which is done. You will also be enrolled for a blood work for CDKL2 levels itself, and that is mostly for research purposes. There is no like therapeutic target for the CDKL2 levels just yet, but that is for the research aspect. But to ensure that the child is doing properly, like, there is no other levels that are being impacted, the kidney function is not impacted, so on and so forth. So for those reasons, there will be the blood work that has been done, and those will be monitored, and the dosing can be adjusted accordingly depending on what the blood work results are.
Okay, thank you. So is this handled by an individual appointment or a day that can be only focused
Yeah,
on?
yeah. So, yeah, so far, I think what we'll do is once we put it on the website for everybody, we'll streamline things so that there can be a one given day of the month where that can be enrolled, or twice 2 given day in a month. So that makes the things simpler and straightforward. All the things can be scheduled on the same day and then done.
That would be really helpful. Okay, sir.
Yeah. We want to do that. So that's why we'll just sort that out and then we'll announce.
And
So the
one more last question from my side. Sorry, Jano,
no
cutting you.
problem.
Yeah.
Yeah. So is this squint eyes is really is this causing because of this seizure? Squint eye
Yeah,
IVs?
it has squint is yeah, it is multiple additional reasons can be there. It because of the visual impairment, they can have a squint, and they can also have neuronal dysfunction. They can squint. Multiple imbalance they also can give rise to squint. So all the things, examination and vision part of it, everything has to be checked.
Okay.
So basically, one of the symptoms of CDKL5 disorder is low muscle tone, which is why the eyes, the squinting is happening, and the muscles of the behind the eyes are weaker, and they're not able to control them, as Dr. Lokesh Lingappa, Dr. Lokesh said that. So, and there are other multiple reasons also, like lack of focus from the brain side because due to cortical visual impairment and so on and so forth. So that correction ability is little reduced as compared to other children who may have squint eyes. The next question, Dr. Lokesh, is are there any long-term side effects of allopurinol medicine? And
No. It is very safe. It's been used for a long time, especially. Yeah, it's used in cancer chemotherapy as a supplement because when you're treating cancer, the cells are dying and uric acid increases. So sometimes people say, oh, why you are using a cancer medication? It's not a cancer medication. To clarify, it is used to reduce the uric acid. When the cancer cells are dying, the uric acid goes up. So that's why it is prescribed in that situation. So it is very safe, can be given at very minimal dosages, usually taken twice daily, and they are very safe medication, and it's been in the market for a very, very long period without any side effects.
Thank you, sir. And the second part to that question was any blood test that I should be concerned of? And I think we covered that, but you can repeat that.
We covered that, yeah.
Yep. Okay. So there's one thing I wanted to
Okay.
highlight.
Yeah.
I'll come to that, San, but just one quick note.
Yeah.
So with respect to allopurinol, in certain Asian population, not specifically Indian, in certain Asian population, it is a contraindication. So for depending on the demographic, there will be an additional test that will be requested before your child can join. And specifically, like, the subset of population that lives in Hong Kong, Singapore, and in that zone, in some regions of China as well. So so it's not like a miracle drug, and like one thing will work for everybody. Each child will be assessed individually for this. Okay, the next question, sir, is regarding dosage, and the question is it 2 times a day?
Twice, yeah.
Yes, it is. Yeah.
Usually around 3 to 5 milligram per kilo. Yeah.
Sorry, sir, go ahead. I was just saying that your voice was cracking a little bit.
Yeah. No, around 3 to 5 milligram per kilo is the usual dosage that is used for other indications, like reducing the uric acid and this one.
Okay. So I don't know how much more longer Dr. Lokesh has, but let's take this opportunity to ask him any and every question we have. Like we are already over 5 minutes, so like he's very generous with his time right now. So please do make use of it before he decides to drop off.
Sir, to refrain the muscle tone, is there any physiotherapy or that can help to refrain the muscle tone? Do you have any other help for that?
See, I think everything has been done actually, as your parents said, no, you have done everything possible. But I think it is always a challenge in this condition because very minimal response is noted. But I think just doing a therapy keeps them in a good shape and not developing contractures and other things. I think that's what the utility of physiotherapy would be.
Okay.
So also, I think the question was specific to like eye muscle tone with physiotherapy. So
no,
eye muscle, is it? Okay.
No, not eye muscle, sir. Regular.
Not eye muscle. That's what I was thinking. Yeah, not eye muscle.
Hand
in general.
muscles and all.
Yeah,
Yeah,
in general, yeah.
in general,
Therapy is
yeah.
definitely,
Yes.
yeah,
So,
they're
Preston, I don't know
beneficial.
if, Mhm. Preston, I don't know
Yeah.
if you attended the previous webinar where, the Napa Center therapist were talking about how to work with muscle tones and what the effects are. So, they did share a few examples of what muscle tone looks like, what is high, what is low, what can be done to help improve it. Obviously, as Dr. Lokesh mentioned, it's not, like it's not going to be miraculously cured overnight, but this is long-term, therapy, and there are ways in which it can be improved. So if you've not seen that webinar, it's already uploaded on the CDKL5 South Asia YouTube channel. So please do visit that. And
Sure.
similarly, for the eye specifically also related to CVI, there were many exercises that were shared in the webinar. So that is also on YouTube. So please, you can
Thank you.
you can check those out. And if there are any questions, you can let us know.
Thank you.
Yeah.
So one thing to add, Janu here and Dr. Lokesh. So like a part of the discussions with the Lovelow Foundation, we were discussing that they are doing the biomarker studies and all that, right? So there's a LPS study that they are doing. We can initiate a discussion with them also that can we participate our community also part of the same study so that we can be part of the larger group itself, and so that that's one thing which we would be taking it up along on that side to explore.
That's a great question, Sumit. So you were at the
No,
conference.
I was just informing that this is one thing which we would be taking it up. Okay.
Oh, nice. Okay,
There's
that's
a,
no,
there's
that's
a
great.
discussion with yeah. So I will include you in that email. I think I missed that earlier, but yeah, I'll include
Okay.
you.
No, that's nice. That's great. Thank
Yeah.
you. Thanks for the
So
update.
would means it would be great that if all of us can participate in that as and when it becomes like if they approve and they are able to kind of extend that for India. So then we are much more into the closer. We can reduce that timeline of 2 years, which Dr. Lokesh did mention earlier, right? So hopefully.
Yeah.
So last 2 minutes, please, ask any questions or any updates that you may have to share. And then at 10 minutes past the hour or 40 minutes past the hour in India, we'll wrap up this webinar for today.
Yeah, I think one of the important thing is wherever the clinicians, I think now as we have discussed, one of the biggest lab in India, genetic lab, we are putting up the website as a part of the diagnostic, this one. Once they put in the diagnosis, the CDKL, the South Asia information will be available so that more and more parents are there. I think it's a parents community coming together in large number, which is what is going to work. That's how it works in US and some of the European countries. So US parents have to drive, because as a clinician, I will be seeing at least in a day, I see 10 different genetic disorders. So as a parent, you are looking at one child who has this and day in, day out. So I think coming together in large numbers is what makes a difference to make it, when you have to talk to the government or policy change and other things. I think each, wherever you, whichever the clinician, you'll have to tell them that we are part of this and this is what is going on. So sometimes not all clinicians are aware. So I think that initiative has to come in so that we can work as a larger group.
With that, thank you so much, Dr. Lokesh, for your time and
Thank you.
this slide deck. It was very informative, very helpful. And for the next steps, we'll put it on the website. We'll put your phone number. If anybody has scheduling challenges or any questions, then
Yes, absolutely.
reach out to you. And with
Yeah.
that, everyone, please have a great rest of your Sunday and a great week ahead. Thank you so much for joining.
Thank you, everyone. Bye.
Thank you, Dr. Lokesh. Thank you, Janu.
Thank you.
Thank you all.